Hi everyone, this little video is a PowerPoint I put together to cover Key Area 3 Photosynthesis Part 2 of the National 5 Biology course, Unit 3 Life on Earth. Now, the Part 1 was done earlier last week and that covered the basics of photosynthesis as well as the first stage of photosynthesis. This video is going to recap what we know from last time, and we're going to go into the second stage, which is carbon fixation, and then we'll look at limiting factors of photosynthesis. Now here's the learning intentions for the whole key area. We're just going to be focusing on the yellow stuff just now. Here are the SQA notes that the SQA have provided that basically say what they want you as a National 5 candidate to know about carbon fixation and limiting factors. You can pause this just now, you could take a wee note, you can maybe add these to your notes. I'm going to be going over this but in greater detail so that hopefully you can understand it a bit better. Before we get started, we're just going to have a little reminder as to what photosynthesis actually is. Now remember, it occurs in plant cells. In particular, it occurs in the chloroplast and it uses a special pigment called chlorophyll. And what happens is we take carbon dioxide from the atmosphere and water from the soil. So the carbon dioxide enters the plant through the stomata, the holes on the underside of the leaves. There are some stomata on the top, but basically holes in the leaf that allow gas exchange. Carbon dioxide enters through the stomata, water enters via the roots of the plants, and then with the help from the energy from the sun, the light energy, we take those reactants and turn that into glucose, the very important molecule that's going to give the plant its energy it needs to survive, and also oxygen, which is a waste product that the plant basically chucks out into the atmosphere through the stomata. As a reminder, this is not just important for plants because all the energy in any animal has originally come from a plant. Okay, so plants take the energy from the sun and produce glucose. For animals to get energy, they must eat plants, or they must eat other animals who have already eaten plants. Okay, so photosynthesis is absolutely vital. Now, in the first video I did on this, and last week, you guys worked on stage 1, which is the light reactions. And that is basically taking the energy from the sunlight, and using it to split up water, H2O. So we split the H2O into oxygen and hydrogen. The oxygen, as I've said, is a waste product, leaves through the stomata, but the hydrogen is, I'll just go get my little pen here. The hydrogen is passed on to the second stage of carbon fixation. And remember, this process of splitting up the water also generates ATP, which, if you remember your stuff from Unit 1, Cell Biology, Key Area 6, Respiration, ATP is an energy storing molecule. Now, both the hydrogen and the ATP are required for stage 2. So, quick recap. Light reactions. We had our water, we've got our sunlight. Okay, the light energy is trapped by chlorophyll in the chloroplast. It splits up our water. Oxygen is released as our waste product, and hydrogen is used in carbon fixation. And during the process, we produce ATP, which also goes forward to carbon fixation. Okay? So, the light reactions are important for giving us the hydrogen and ATP required for carbon fixation. Now, the carbon fixation stage is all about taking that hydrogen and fixing it with carbon and oxygen to make glucose. Now, this molecule here is our glucose molecule, and as you can see, it just contains carbon, hydrogen, and oxygen. Now, right now, from the light reactions, we have the hydrogen that we need, but we don't have the carbon and the oxygen. So that's when the carbon dioxide, the other reactant we haven't talked about yet because it's not used in the light reactions, that's when that comes in from the air and the carbon dioxide joins with the hydrogen using the energy that was made earlier, that ATP, to produce our glucose. Let's go over how that is done. So, from light reactions, we've got hydrogen. From the air, we've got carbon dioxide. We bring those two together with the help of ATP from the light reactions, and we produce glucose. Okay? Now, I want you to look here. Our ATP is turning through this process into ADP plus PI. Now remember what I said, ATP stores energy. So for us to combine our hydrogen and our carbon dioxide, ATP breaks up, so it basically chucks off that last phosphate, releases the energy that it's storing, so that we can combine the hydrogen and carbon dioxide to make glucose. Now because that phosphate has been taken off the ATP to allow the cell to access that energy, we now get ADP plus PI. Okay? So we're left with glucose. We already had the oxygen as a product. That left a while ago as a waste product from the light reactions. Now we've got our second final product, glucose. Now a few things can happen to the glucose. The glucose can be used immediately by the plant as a source of energy in respiration. Remember, respiration is the breaking up of glucose to release the energy inside glucose. And cells do this so that cell activities can occur, like protein synthesis, mitosis, which is cell division. Any activity inside the cell that requires energy relies on respiration. Now, if the cell doesn't need the energy immediately, it's not going to go to respiration. It could go and form a storage molecule called starch. And that's how the plant will store its energy for later. And if they ever need the energy, basically the starch will get converted back into glucose, and then it will go forward for respiration. So starch is a store of glucose. So glucose is a simple carbohydrate, but if you combine it together, it makes starch, which is a more complicated carbohydrate, and that's how it's stored. Something else that could happen to glucose is it's going to be converted into another carbohydrate called cellulose. Now hopefully you're remembering that cellulose is found in the cell walls of plant cells. That's taking us straight back to Unit 1, Key Area 1. The first thing you looked at was structures of the cell. Now a plant cell wall is made of cellulose. So if the plant is undergoing mitosis, which it will be, it'll be producing new cells, it will need some of that glucose to form cellulose so it can produce more cell walls. Throughout this whole process, there's multiple reactions happening, and all of them are controlled by enzymes. Again, don't know what enzymes are? Go back, Unit 1, Key Area 4. Enzymes are special proteins that speed up chemical reactions. We've got enzymes at the start. We've got enzymes to the middle. We've got enzymes at the end. Okay? Enzymes are very important, and that's going to be an important point later. Right, recap those uses of glucose. I just mentioned them. The glucose can be used in respiration. It can be stored as starch. Now, look at this diagram here. Unfortunately, we can't do this experiment just now because, well, you're learning from home, and a lot of us won't have access to iodine solution, okay? But we did this in S1 or S2. If you take a plant that's been kept in the dark, and then you take a plant that's been kept by the window, if you do the starch test, so basically you're going to add iodine to the leaf from the plant that's been in the dark and the leaf from the plant that's been by the window, you'll be able to see that one of those leaves has more starch in it, because iodine is normally an orangey colour, orangey brown. It turns blue-black in the presence of starch. Hopefully this is ringing bells, and what that shows you is basically the plants that have done more photosynthesis have more starch because they produce more sugar, so they're using it as a store. Hopefully that's ringing some bells. And again, the other thing that we can do, as I mentioned, cellulose. In this diagram, we've got a picture of a kind of cross-section of the cell wall, and you can see that in the middle of the cell wall is these cellulose fibers. So that's another use of the glucose. It gets turned into these cellulose fibers. Now, Wee summary for carbon fixation. It's a series of enzyme-controlled reactions. That's a buzzword, a buzz phrase you need to know. It uses hydrogen ATP from the light reactions and carbon dioxide from the air. The carbon dioxide and hydrogen combine using energy from the ATP to produce glucose. The glucose could be used for respiration, conversion to starch for it to be stored for later use, or converted to cellulose to provide structure to the cell walls. So that concludes the actual process of photosynthesis. Last week was stage 1, this week was stage 2. So now we know how photosynthesis works. We now need to discuss something called limiting factors to photosynthesis. And this is the final bit of this key area. Okay. Now, a limiting factor is basically something that can affect the rate of an activity. I'm going to use this metaphor about a factory line first before we apply it to photosynthesis so we can really understand what's going on here. So in this diagram, we've basically got a machine that is producing CDs. And there are 3 different materials that go into the production of this CD. Okay? So you have to imagine that you need these 3 materials to produce a CD. Now, if you don't know what a CD is, which I don't think I'm that old or you're that young, but CDs are basically what people use to play music on. Now you probably use MP3s and whatever. I'm now going to sound really stupid as I try and describe what you use, but there's little machines and you put it in. Anyway, we used to have to use CDs. So we're imagining that we need these 3 materials to make a CD. Now, imagine that the red material is harder to find than the blue or the green one. Okay? So, even if we've got lots of blue and lots of green, we can't make lots of CDs. We can't make CDs really quickly because it relies on all 3. So that means in this incident, the red material is limiting the rate at which we can produce CDs. So it's a limiting factor. I mean, it doesn't have to be red. It could easily be maybe I don't know, further down the line suddenly there's a crash in the production line for this green material, and now it's even harder to find the green material. Then it'll become the green material is the limiting factor. Okay, so basically the limiting factor is anything that is required to produce something, but it's in short supply. As soon as it's not in short supply anymore, it's no longer a limiting factor. Okay? So let's have a look at photosynthesis. If this is our word equation for photosynthesis, carbon dioxide plus water through the action of light makes glucose and oxygen, what could be a limiting factor of photosynthesis? Okay? Now, I want you to pause this and try and come up with it. What I will tell you is there are 3. The SQA does not accept water, okay? So water is not counted as a limiting factor. So I want you to pause this and see if you can come up with the 3 limiting factors. Okay, hopefully you've unpaused this now. Limiting factor number 1 will be carbon dioxide concentration. Now, carbon dioxide could be a limiting factor because it's needed for photosynthesis. So, if we limit how much carbon dioxide is available, we're going to limit how much photosynthesis can actually occur. Number 2 could be light energy, or light intensity, sorry. So, if light energy is limited and there's a low light intensity, that means photosynthesis is going to be occurring at a low rate, because light energy is required for photosynthesis. Okay, see where this is going? So if it's required for photosynthesis, it could be a limiting factor. But just remember, not water. SQA doesn't like you saying water. I don't know why. But they have their reasons. I'm sure they do. However, I said there's 3. And if the third one's not water, then what is it? Now, enzymes control the reaction of photosynthesis. What do we know about enzymes? Well, if you think back to Unit 1, KA3 you looked at factors that affect the activity of enzymes. So hopefully you'll remember that Temperature affects the rate of activity. So in cold temperatures, there's not much photosynthesis occurring. This is why you don't need to cut your grass in the winter, because it's not growing much, it's too cold. Whereas in the summer, where it's warmer, you need to cut your grass more often, because those enzymes are starting to work and starting to actually get that grass growing, because photosynthesis is occurring. Right, you need to be able to look at these graphs. Now, here is the, sorry, checking my time, I've got a meeting. Here is the graph for light intensity. Now what we've got up the side is rate of photosynthesis. So if we start at the bottom, that's a low rate and this is a high rate. And again, we've got light intensity along the bottom. So this one is low light intensity and that is high light intensity. So what this graph says, it says that as light intensity increases, so does the rate of photosynthesis. Up to a point, okay? Then it starts to level off, right? Same things happens with carbon dioxide. As the concentration of carbon dioxide increases, the rate of photosynthesis increases, but then it starts to level off. We're going to have a look at that to explain that. And now temperature, same thing again, or is it? It's not. Okay, so as we increase temperature, we increase the rate of photosynthesis up to a point, and it doesn't level off, it now starts to decrease. Okay, so we're going to have to think about why that is. Hint, hint, it's linked to enzymes. Right, let's look at carbon dioxide first of all. At low concentrations, the rate of reaction is limited by how much carbon dioxide is available. So as you can see, as we increase the carbon dioxide, we are now increasing the rate of photosynthesis. Okay? But then it starts to level off, and it's because carbon dioxide, at the point that it levels off, is no longer the limiting factor. So at this point, carbon dioxide isn't limiting the rate of photosynthesis, because as carbon dioxide increases, photosynthesis doesn't increase. If carbon dioxide was the limiting factor, then the line would have to continue to go up in a straight line, not a squiggly line like that. This means because it's leveled off, there must be something else limiting the rate of photosynthesis. Carbon dioxide is no longer the limiting factor. So what could it be? Light intensity or temperature. Now if we look at light intensity, we have the exact same situation. As light intensity increases, we increase, and then it levels off. So again, at this point, at the point it levels off, light intensity is no longer limiting the rate of this reaction. So what could it be? Well, for this section, it might be limited by carbon dioxide or temperature. Okay? So the relationship between light intensity and the rate of photosynthesis, and the relationship between carbon dioxide and the rate of photosynthesis is similar. Okay? It goes up and then levels off. Because when it levels off, it's no longer limiting it, something else is the limiting factor. Let's have a look though, because temperature is doing something different. Okay? Here we've got a graph of temperature, right? So temperature is increasing along the bottom, and then we've got the rate of photosynthesis increasing, oh gosh, up the side. My arrow went a bit crazy there, didn't it? And what we can see is, as we increase temperature, the rate increases up to a high point, and then as we continue to increase temperature, the rate of photosynthesis decreases. Why? Okay. Remember, enzyme-controlled reactions, so enzymes are important. At high temperature, enzymes denature. If you don't know what that means, and this doesn't ring any bells, Unit 1, Key Area 4. So remember that an enzyme is a protein, and this green bit here signifies the protein, and a substrate is the thing that basically goes into the active site like a lock and key and allows a reaction to take place. So these enzymes and substrates are important for photosynthesis. However, if we increase the temperature too much, the amino acids that are joined together to make our enzyme, the bonds between them start to break. So it starts to denature. So now the active site takes on a completely different shape. So the substrate can no longer bind. So the reaction can no longer occur. So in terms of photosynthesis, it's going to slow down and stop. So you need to be able to describe the differences between the graphs of our limiting factors: carbon dioxide, light intensity, and temperature. Let's have a little summary. Okay. Limiting factors is something that can affect the rate of photosynthesis. They include light intensity, temperature, and carbon dioxide concentration. If any of these factors are in low supply, the rate of photosynthesis will be reduced. It's going to be low because the chemical reaction needs these things. If you increase the limiting factor, the rate will increase until it's no longer a limiting factor, and then something else will be the one that's limiting the reaction. But if you increase temperature above its optimum, so this peak here, photosynthesis will then start to reduce. Right. That's us finished off key area 3. Hopefully that's helped you sort of understand what's going on, listening to me talk through the slides. Obviously there's going to be more activities for you to kind of consolidate your learning and bring together the stuff you learned last week with this week. If you've got any questions, just ask, tag me in Teams. If you're actually here by chance and you've got nothing to do with my school and you've got any questions, just ask in the comments, and please like this video if you found it helpful. Okay, right, see you guys later. Bye.